support the growth and viability of these yeasts. Another way of looking at this is by expression strength. Now you can do a similar experiment to comparing the 30 degrees to 16 degrees. You can do it all at 30 degrees, again optimal temperature, but you can vary the expression of HSP90 like I showed in the original slide panel on the left of the first plot. So you can see if you look at the wild type, which is in the green box up at the top, pretty much you can reduce HSP90 expression to less than 10% of normal. And the yellow color shows that the yeast are able to tolerate that and are growing fine. If you look at the W585T mutation, which is shown in the red box, you can see that at 30 degrees at optimal concentration, like I showed you in the previous slide, the yeast are happy. But as you begin to decrease expression, even though growth conditions are optimal, it begins to not work and to not support yeast growth. So the reason I'm showing you all this is that the bottom line is that in yeast as well as in human cells, there's generally an excess, a large excess of HSP90. You can reduce it a lot and have no obvious effect. But if you stress the yeast, either by giving them an HSP90 that is not completely active, or if you put them at a temperature that they don't like, they now become very dependent on optimal expression. And one can imagine a similar situation for cancer cells. So the green line is showing what would happen to a normal cell if you reduce HSP90. You'll get to a point where it's going to have an effect because HSP90 is essential for growth. But normal cells don't need all of the HSP90 that they have. In normal cells and tissues, HSP90 makes up somewhere around 3% of the total protein of the cell. So it's quite a lot. It's in micromolar concentration in cells. And they can get by with a lot less. But if you stress the cell, or instead, if you look at a cancer cell, which as I'll show you in a minute, is essentially stressed all the time, then they can tolerate only a little reduction in HSP90 levels before you see significant effects on growth and viability, or fitness, as shown here on the y-axis. Now, it's not exactly correct to equate expression with inhibition, but an analogy can be made suggesting that you can inhibit HSP90 to a sufficient degree where you're going to see an effect in cancer cells while you see minimal impact on normal cells. And over the years in published papers, this is in fact what we see. So cancer cells in general tend to be more sensitive to pharmacologic inhibition of HSP90 compared to normal cells. And a likely reason for this is that HSP90 is one of the main response mechanisms that the cell and the organism has to environmental stresses, some of which that are common to cancer are shown here. On the left-hand side, cancers usually outgrow their blood supply, and so nutrient stress is a common factor. There's proteotoxic stress because there's usually a lot of reactive oxygen in cancers. They're hypoxic, so they're not getting enough oxygen to have the mitochondria function at optimal activity. And many, if not most, cancers are aneuploid and are characterized by extreme genetic instability. This leads in turn to the synthesis of mutated proteins that are not able to fold and must be dealt with by the cell, either to chaperone them until they're folded or to target them for degradation. And HSP90 is involved in both of those processes. So all of these stresses impinge on HSP90, and HSP90 in turn controls the fate of a host of client proteins that are signaling proteins, for example, the one shown on the right-hand part of the slide, that mediate a lot of the processes important for cancer cell growth, survival, metastasis, etc. The gray boxes describe what are now accepted as the hallmarks of a cancer cell that distinguish cancer from normal. And you can see that all of the hallmarks that are shown here, there are client proteins that depend on HSP90 that are important for each of them. So that you would think that by targeting HSP90, you can have a major impact on the viability of cancer cells in vitro and in vivo. And a correlate of this would be to suppose that if you stage cancers by the amount of HSP90 they have, the more HSP90 expressed in a cancer, the more aggressive the cancer might be, and the worse the survival or the prognosis of the patient would be. And this, in fact, has been shown by looking at tumor tissue that's taken from patients for a number of different kinds of cancers, including non-small cell lung cancer, which I'm not going to show you today, but also in breast cancer, which I'm showing you on this slide, where if you stratify the tumor samples from patients with triple-negative breast cancer and make a cutoff where above that is arbitrarily called high HSP90 and below that is low HSP90, you can see that in tumors where HSP90 is upregulated significantly compared to the remainder of the population, percent survival is markedly reduced in the patient. So expression level of HSP90 is a negative prognostic indicator of cancer aggressiveness and survival. It's a prognostic indicator of aggressiveness. It's a negative prognostic indicator of survival. So our idea was that inhibiting HSP90, like knocking down expression of HSP90 in simple systems like yeast, should compromise cancer cell robustness. And this, in fact, turns out to be the case. This is an experiment we published a long time ago now that shows that this prototypical HSP90 inhibitor, 17-AEG, which is the first in-human HSP90 inhibitor, it is very effective as a radiosensitizing agent. We're looking here at a couple of lines, HeLa cells and CHA cells, and you can see the doses of irradiation on the x-axis and the surviving fraction on the y-axis, and the y-axis, make note, is in a log scale. So you can see that at this dose of radiation, the HeLa cells and the CHA cells, which are both lines derived from cervical cancer, are sensitive, but certainly there's a significant fraction of cells that remain after irradiation. If you add in low doses, these are nanomolar concentrations of this HSP90 inhibitor, you can see that you markedly improve the toxicity of irradiation, and this definitely is combinatorial because in the absence of radiation, this dose of HSP90 inhibitor isn't doing anything by itself. This effect is time dependent. If you look at the panel on the right, you can see that the longer the pre-exposure to HSP90 inhibitor, the better the sensitizing effect, going from 4 hours pre-exposure, which essentially has no sensitizing effect, to 12 hours pre-exposure, which has a significant effect. You get lots more cell killing. So far, the data suggests, and